I'm Bob Banks of Durham University in the UK. This short video is about the editor's pick in the special issue of Experimental Physiology on mechanotransduction, muscle spindles and proprioception. I was first introduced to the mammalian muscle spindle over 50 years ago as a graduate student in Sheffield, and I have been studying its structure and function ever since then, mostly in Durham. The spindle is not as well known in general as it deserves to be, since we each have more than 50,000 of them in our skeletal muscles, and they're essential for the control of posture and locomotion. Each is a complex sense organ in which specialized muscle fibers, known as intrafusal fibers, receive both sensory and motor innervation. The whole thing is enclosed within a multi-layered capsule and lies alongside and among the much larger force-producing fibers that make up the great bulk of the muscle. There may be several sensory endings in a single spindle, each supplied by its own axon, but one in particular is essential for the initial formation of the spindle and is therefore found in all spindles. It is called the primary ending, and its sensory terminals are distributed to all of the spindle's intrafusal fibers. The axons supplying primary endings are the largest and fastest conducting in the body. For most of their lengths, intrafusal fibers have a sarcomeric structure similar to all skeletal muscle fibers, but where they are innervated by the primary ending, almost all the sarcomeric structure is replaced by large, closely packed nuclei. When I began my work on spindles, it was widely accepted that there were two different types of intrafusal fiber. One was larger with a bag-like cluster of nuclei, and the other was smaller with nuclei in a single row. Inevitably, they became known as nuclear bag fibers and nuclear chain fibers. It was also known that functionally, there were two types of motor nerve fiber, supplying spindles and ending on the intrafusal muscle fibers, whose axons could differentially modify the response of the primary sensory ending to stretch. The simplest model to combine and account for these observations was to assume that the two types of motor fibers separately and specifically innervated the nuclear bag and nuclear chain fibers. The problem is that simple models are often wrong, and this was no exception. Some of my earliest work as a PhD student was to provide clear evidence that there were not two but three distinct types of intrafusal fiber, a conclusion that we confirmed after I joined David Barker's group in Durham as a postdoc. This necessitated a re-examination of the innervation of the spindle, especially the two functional categories of muscle fibers. How this was done and the conclusions reached occupies much of the first retrospective part of There and Back Again, 50 years of wandering through terra, incognita, fusorum. In the second part, I describe some new observations on the geometrical properties of the sensory nerve terminals and the underlying nuclei of the intrafusal muscle fibers. Clusters of large, euchromatic, closely packed nuclei are a unique feature of the spindle sensory region. It seems highly unlikely, therefore, that so many nuclei are required for large-scale gene transcription in such a small space. Rather, I am suggesting that they play a mainly mechanical role as three-dimensional springs, contributing to the elasticity of the sensory region. For the last 20-odd years, most of my work has been carried out in collaboration with Guy Buick of Aberdeen, and we are studying the processes of mechanosensory transduction by the spindle's sensory endings. My new observations on the sensory terminals and the associated myonuclei are a small contribution to our understanding of the transduction process.